Hello everyone, welcome to E-PG Pathshala. I am Dr. Veda Krishnan. I am a scientist in the Division of Biochemistry, Indian Agriculture Research Institute, IARI, New Delhi, India. I am going to talk today about a very relevant topic, that purification of enzyme part 3 from the paper Protein Biochemistry and Enzymology. In today's lecture, we will focus on understanding the methods for qualitative and quantitative estimation of protein, which are essential during each step of a protein purification process. These are like day-to-day lab protocols. For example, we will look at assays for determining protein content in a sample and also study the electrophoresis of protein in detail. To start, this concept map shows the outline of today's lecture. We will first study the various protein quantification methods, which include the protein assay and determination of specific activity and yield. In the next session, we will cover SDS-PAGE in detail, spanning an understanding of the gel metrics, the role of SDS and buffer system, the electrophoresis, sensitivity of SDS-PAGE, application and the staining technique like Cumacy, silver staining and so on. We will also cover isoelectric focusing and 2D electrophoresis briefly in this module. The quantitative estimation of protein is a very important and necessary step throughout the entire purification process. The knowledge of the amount of protein present after each step in purification is an essential criteria. Depending on the amount and purity of the protein available, different chromatographic methods can be selected for protein separation. The various methods of estimating protein quantitatively are shown in this figure. Measurement of the protein absorbance at 280 nanometer is one of the most simplest and direct assay method for protein present in solution. Look in this figure. Amino acid containing aromatic side chain like tyrosine, tryptophan and phenylalanine exhibit a strong UV light absorption at 280 nanometer. Thus, proteins and peptide will absorb UV light in proportion to their aromatic amino acid content and concentration. Several colorimetric protein assay techniques are available, which are used routinely for determining the protein concentration. The principle of these colorimetric approaches is that when protein is added to the coloring reagent, a complex is formed leading to a color change, which can be read colorimetrically and which is proportional to the concentration of the protein added. A standard curve is a must, consisting of the concentration of purified reference protein is plotted previously to assist in the determination of protein concentration. The various colorimetric methods used commonly for protein estimation are Biuret method, Fall-in-Lowry method and Bradford method. The Biuret reagent comprises of hydrated copper sulfate together with potassium sodium tartrate in an alkaline medium of sodium hydroxide. When mixed with protein, the cupric ion complexes with peptide nitrogen in protein to generate a pink or violet blue color complex as in this figure. The coloration of the complex is proportional to the concentration of the protein and can be read spectrophotometrically at 550 nanometer. Let's see what are the properties of Biuret method. It's a very commonly used method because it is not affected by any contaminating amino acid, since the complex formation depends on at least a dipeptide structure. It is also not interfered by the presence of nucleic acid. But the major disadvantage are this test can be interfered by some compounds like ammonia or amine. Thus, false positive result can be obtained if ammonium sulfate salt or tris hydroxymethylaminoethane salts are present in the sample. Another issue with this method requires a high concentration of protein in the range of 5 to 160 mg per milliliter. The Lowry method is a modification of already discussed Biuret method. After treatment with cupric ion, the protein is treated with Folin-Kayakal reagent, which is a mixture of phosphotungstic acid and phosphomolybdic acid under alkaline condition. Look at this blue color in this figure. It is the product of this reaction, heteropolymolybdenum blue. This absorbs strongly at 750 nanometer. Let's see the properties of Follin-Laurie method, another commonly used for routine assay. It is more sensitive and can detect lower concentration of the protein from 0.01 to 1 milligram per milliliter. The assay as well as the standard curve must be performed at lower protein concentration since the linear dependence is achieved only at low protein concentration. But as it is a modified biuret method, it is sensitive to the presence of contaminants, and many more. The assay need to be performed with high precision. The basic principle of this assay is that a dye named Cumasi brilliant blue binds to the protein under acidic condition to form a blue colored complex that absorbs strongly at 595 nanometer. This method is different as it is independent of the amino acid composition. A standard curve and dilution of the sample is always essential for this method because the assay is linear for a very short range of protein concentration, 0 microgram per milliliter to 2000 microgram per milliliter. This assay also interfered by the presence of detergents like SDS. Careful handling is very important because the dye stain not only the protein but also cuvette, test tube, floor, etc. This must be taken care that the cuvette and test tube stain can be removed by washing with ethanol. The success of a protein purification scheme is also evaluated by determining specific activity and purification yield after each purification step. Let's see. First is total protein. The total amount of the protein present in a fraction is obtained by determining the protein concentration present in each fraction. This is obtained using one of the protein quantification method stated above. Bradford method is most widely used. Its unit is usually in milligram or microgram. Coming to activity. The activity present in any fraction is obtained by measuring the enzyme activity using assay specific to the enzyme. The total activity can be obtained by adding the activity in each fraction. Its unit is usually expressed as U. What is specific activity? Specific activity is a parameter which gives us an idea of the purity of the enzyme in the mixture. It is the activity of enzyme per unit mass of the protein obtained by dividing the total activity by total protein. Its unit is U per milligram. The purer the protein, the higher is the specific activity. Coming to yield. It's a measure of how much activity is retained after each step in purification. The enzyme activity retained after each purification step is divided by the total initial activity of the crude extract and is expressed in percentage. Purification fold. It is an index of how much the specific activity of the target protein has increased in comparison to the initial specific activity when the target protein was present in the crude extract. It is obtained by dividing the specific activity by the specific activity of the initial crude extract. It has no unit. Higher the purification fold, greater is the extent of protein purification that has been achieved. An efficient and good purification scheme should take into consideration both the purification fold as well as the yield. Next, we will go into studying SDS-PAGE electrophoresis. SDS-PAGE, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis, is an important approach to analyze how much purification strategy has been through visualization of the protein present after each step of purification. The technique is based upon the principle that SDS denatured and negatively charged protein molecule migrate towards a positive electrode in the presence of an electric field. In the presence of an electric field, the protein in the gel migrate with different migration rate based on the mass, and simultaneously they get separated. Electrophoretic separations are mostly performed in gel which serve as a molecular sieve. A typical setup for SDS-PAGE electrophoresis you can see in this figure. The electrophoresis is performed in a thin vertical slab of polyacrylamide gel formed by the polymerization of acrylamide and cross-linked NN-methylene bisacrylamide. Protein molecules that are smaller in size as compared to the pore sizes in the gel can readily move through the gel, whereas molecules size larger in size than the pore travel comparatively slowly. Intermediate size molecules move through the gel at an intermediate rate. The direction of the movement of the protein in the gel is from cathode to the bottom anode. When proteins are dissolved in solution of sodium dodecyl sulfate, that is SDS, and further boiled, the secondary and tertiary structure of the native protein are completely disrupted, like you can see in this figure. Mercaptoethanol or dithiothritol is also added further to reduce the disulfide bond. The denatured protein is now unfolded into a linear structure. Anion of SDS bind to these linear chain at a ratio of about 1 SDS anion to every 2 amino acid residue. This SDS protein complex now has an overall large net negative charge that is proportional to the mass of the protein. This negative charge conferred by the SDS molecule is usually much greater than the charge on the native protein, thus rendering the native charge on the protein as insignificant as you can see in this figure. The SDS-PAGE electrophoresis functions through the use of a discontinuous lamellae buffer system, which implies that different buffers are used in gel and tank. The stacking gel is buffered by tris HCl to pH 6.8, the running gel buffered by tris HCl to pH 8.8, and a tank buffer is buffered at pH 8.3. Have a look at this figure. This figure here outlines the different charge state of glycine, positive, neutral or negative, under different pH condition. The migration of ion and protein during SDS-PAGE electrophoresis is explained below. At the start of SDS-PAGE protein electrophoresis, the chloride ion are in the stacking and dissolving gel and negatively charged glycine is present in the tank. When the electric field is set up, the negatively charged glycine ion enter the stacking gel from the electrode tank buffer. In this environment of pH 6.8, the glycine molecule become zwitterionic in nature. With no net charge on them, the glycine molecule move very slowly in the electric field. On the other hand, the chloride ion from tris HCl move much rapidly in the electric field, resulting in the formation of an ion front that migrate ahead of glycine. A steep voltage gradient is created, resulting in two narrowly separated front of migrating ion, the highly mobile chloride front followed by the slower zwitterionic glycine molecule front with protein in the stacking gel, having an intermediate mobility between these two. The protein are thus stacked into a thin layer between the leading chloride ion and the trailing glycine ions. At the running gel pH 8.8, the glycine molecule become negatively charged and can now migrate faster, and thus they accelerate past the protein. The protein remains stacked in a narrow band at the interface of the stacking and running gel. Further, since the running gel has an increased acrylamide concentration as compared to the stacking gel, the pore size in the gel are still smaller and the movement of the protein is further slowed down. Higher molecular weight protein move very slowly through the porous acrylamide gel compared to lower molecular weight protein. Thus, separation of the protein begins according to their size. The whole principle has been shown in A, B and C figures. When the electrophoresis is complete, the protein in the gel can be visualized by staining them using Cumacy blue dye as figure 13 or silver staining as figure 14, which reveals a series of bands. What is Cumacy staining? The electrophoresized SDS gel is visualized by staining with a dye such as Cumacy brilliant blue R250. The principle of Cumacy staining is the same as that of Bradford protein assay, wherein the Cumacy dye react with the protein to generate a blue colored complex. Figure 13 shows a Cumacy stained SDS gel after electrophoresis. Coming to silver staining, Another method for staining the electrophoresis gel is the silver staining method. Silver staining is used in case when the protein sample loaded into the gel have low concentration and a higher sensitivity is when required. This method of staining, however, uses simple and cheap reagent and equipment. The various steps involved in silver staining of an SDS-PAGE electrophoresis gel containing protein are fixation, sensitization, silver impregnation and development. However, silver staining requires a lot of carefully handled steps. A silver stained SDS-PAGE gel is shown in figure 14. SDS polyacrylamide gel electrophoresis method for protein visualization is a rapid and sensitive method which also provides high degree of resolution to the protein sample which is being separated. While in case of Cumacy staining, the protein with a concentration in microgram range, that is as little as 0.1 microgram, gives a distinct and visible band. The sensitivity is even higher in case of silver staining, where protein sample in the concentration of nanogram range can be detected. Also, protein that differ in mass about 2% can also be separated and distinguished on an SDS gel. SDS-PAGE electrophoresis can be used for the determination of purification efficiency and protein molecular mass. What are the applications? The main application is the determination of mass. The mobility of most protein in SDS-PAGE is inversely proportional to the algorithm of their mass, which can thus be determined. What about the efficacy of purification? The efficacy of any purification scheme can be analyzed by SDS-PAGE. Of the protein eluted as fraction, the initial fraction will display dozens to hundred of protein. As the purification progresses, the number of bands will diminish and the prominence of one of the bands should increase. This band will correspond to the protein of interest. Isoelectric focusing refers to the separation of protein electrophoretically on the basis of their pI, that is, on the basis of their relative acidic and basic residue. The isoelectric point of a protein is the pH at which its net charge remains 0. Therefore, the electrophoretic mobility of the protein at this pH will be 0, since the protein will not be able to migrate in any electric field. When a mixture of protein is subjected to gel electrophoresis across a pH gradient, each protein will tend to move in the gel until it reaches a position where the pH becomes equal to the pI of the protein. At this position, the migration of the protein will stop. Figure 15 outlines the method of isoelectric focusing for the separation of protein. Two-dimensional electrophoresis is an advanced method of protein separation where the separation of the protein on a gel is conducted in 2 dimensions. It involves the combination of isoelectric focusing with SDS-PAGE to obtain protein separation with a very high resolution. The sample to be purified is first subjected to isoelectric focusing as described in the previous session. For the second dimension, the gel is then subjected to SDS-PAGE electrophoresis. The protein now move through polyacrylamide gel vertically based on how far they migrated during isoelectric focusing, as in figure 16a. Post the electrophoresis, the gel is further stained by Cumacy or silver staining to yield visible two-dimensional pattern of spots as you can see in the figure 16b. Thus, in 2D gel electrophoresis, the protein are separated horizontally on the basis of isoelectric point and vertically on the basis of their masses. The protein separated by 2D electrophoresis can further be identified by coupling with mass spectrometric techniques. So, dear students, to summarize, protein quantification is an essential step at each purification step, and protein estimation assay like Bradford, Lorry and Biuret help in the determination of the protein content in any purification scheme. Specific activity and purification yield are very important parameters to be considered at each purification step. Purification table gives an overall understanding of the efficiency of the purification protocol. SDS polyacrylamide gel electrophoresis has emerged as a very important technique to analyze the effectivity of the protein purification and visualization of the protein present at each purification step. It has also been covered in detail in this text. Advanced method of protein separation like isoelectrofocusing, where the protein separation is done on the basis of isoelectric point, and 2D electrophoresis, where isoelectric focusing is combined with SDS-PAGE to obtain a very high resolution protein, have also been studied. In nutshell, in this lecture, we have looked at the various methods for quantitative and qualitative estimation of protein, which give important information about the extent to which a protein has been purified, while the quantitative methods give an idea of the purification fold and increase in specific activity. Electrophoresis method like SDS-PAGE, isoelectrofocusing and 2D electrophoresis contribute towards efficient protein separation. Thank you.